Hey folks, and welcome to another edition of Zog Science. Today we're going to be finishing up our talk about chemistry by talking about carbon and why it's important when we talk about biochemistry. It's important because biochemistry is carbon based. Why is carbon what makes up all of pretty much biochemistry? Well, it's got four valence electrons, and each of those four valence electrons can form up to four covalent bonds. There can be single bonds and double bonds and triple bonds mixed in there. So basically, we can have a lot of variation. We can form a lot of different things. Carbon is also super abundant. It's found, all over the place. So the fact that it's both abundant and it can form four bonds basically makes it the primary thing that starts organic molecules, right? And we call carbon compounds organic compounds. This is not like organic, like it's grown, without pesticides or things like that. When we talk about organic, we're talking about the fact that it is made up of carbon and therefore is most likely got some sort of living thing. An example of a molecule that has carbon in it but is not organic would be carbon dioxide, right? Carbon dioxide would not be considered an organic compound. It would be considered an inorganic compound. One of the main things that carbon can do is because it can form these four bonds, it can create lots of different shapes. And carbon is really good at binding with itself and being very, very stable, even when it is in extremely long chains. And so what we can get is we can get a lot of variety of different things. We can get very long branches. We can also get double bonds in there that mix things up. We can get different types of branching. So we can get ones where the carbon is kind of coming off at an angle as opposed to the main chain. And we can also get rings. And the rings allow for a variety of structures. And we're going to see a lot of these things repeated as we kind of get into our different macromolecules, which are all organic compounds. Now, one of the cool things that we can do is because carbon has these four bonds, we can get a lot of different variety in terms of where the carbon or functional groups, which we'll talk about in a moment, can be attached. And these different configurations are called isomers. So basically, it's the same chemical formula, right? So if we take a look at the example here with the structural isomer, we've got 5 carbons, and then there's going to be 10 hydrogens arrayed around it. Is it 10 or is it 12? Sorry, it's 12 hydrogens that are arranged around it. Depending on how the carbons are bound together, we can get different structures, right? Well, this kind of goes back to what we were talking about before, the relationship between structure and form. Yes, it might be the same formula, but because it has a different structure, it's going to have a different function and have different properties. There are a lot of different ways that these isomers can be built. We can also have cis or trans isomers. This is where we've got a double bond, and the functional groups that are on either side of the bond are flipped and on different sides. Again, it's the same chemical formula, the same elements are found inside of it. However, they're arranged differently, therefore it has a different property. And then finally, we have what are called enantiomers. And this is where we have a carbon that has different things bound to each of the sides. And those can either be what we call left hand or right hand. And those left and right hands basically have to do with whether or not those molecules are attached like clockwise or counterclockwise. And that's very important because how they're attached, again, is going to impact their structure and their function. All proteins are made up of L amino acids, and we also only use D sugars when we are looking at organic compounds. And this is another one of those things that helps us to kind of look at and see that life is all coming from a single original common ancestor, because it makes sense that if our common ancestors only used L amino acids, that all of their offspring would. And so we find throughout all of life that we've got L amino acids and D sugars. Now, I'm sorry, maybe not necessarily through all of life, but within groups of families, groups of organisms, you've got L amino acids and D sugars, at least for us. For others, it can be different. Now, functional groups. What I was talking about before when I mentioned functional groups is basically we have these kind of discrete packets of compounds, elements that bind in places, and they have specific properties that they confer onto into the overall compound. So, for example, the hydroxyl group is an OH, and it generally is going to attach onto a carbon somewhere. And this is going to be an alcohol. So every time you've ever heard of like ethanol or methanol or something like that, these are all alcohols, and they all have a OH group that is bound to the carbon chain. Propane is an example of this, or octane, right? These are all hydroxyls as well. Sorry, that is not true. They're not hydroxyls. Those are just chains of carbons. But Because we've got this hydroxide on the end, it makes the molecule polar, which we kind of talked about in class. And this is going to allow it to be able to form hydrogen bonds with water molecules, helping it to dissolve. And it allows things to dissolve easier, which is good for things particularly such as sugars and things like that. The next functional group is the carboxyl group. The carboxyl group is also known as the carboxylic acids or organic acids. And what it has is it has a double bond oxygen with a hydroxide on the end. And what that does is that it allows it to donate this hydroxide, making things acidic. And so one of the example of that is the acetic acid, and acetic acid is what gives vinegar or lemons their sour taste. And so again, this is very important. We're going to see the carboxylic acid group a lot when particularly when we take a look at proteins. The next one is the amino group. The amino group is found on amino acids. So for example, you'll notice here that this is glycine, which is an example of amino acid. It has an amino group on one end and it has a carboxylic acid group on the other. And so what the amino group has is that it can act as a base and it can pick up a hydrogen and therefore it is going to help make the things around it more basic. So it's very important when we start talking about proteins, the interactions between the carboxylic acid groups and the amino groups on either side, one being acidic, one being basic. Final group is the phosphate group. The phosphate group is going to contribute negative charges because it's got some oxygens that have extra lone pairs of electrons. And phosphate groups are very important because we find them in DNA. And they are part of the sugar phosphate backbone. And so it's very good to have a basic understanding of the phosphate group and kind of what it is capable of. One final thing to mention about functional groups is that the placement of functional groups is very important to the function of a molecule. Okay, so what we have here is we have estradiol, which is a type of estrogen, and then we have testosterone. Right, these two molecules have very different functions. Right, one of them is found primarily in females, one of them is found primarily in males, and they have a lot of things that they do within those two genders, sexes. But they do different things, right? And that all has to do with the fact that we have slightly different functional groups on each of them. You notice that we have the same general structure here, and this is actually the structure of a steroid. But the difference here is that they've got these different functional groups on either end. And so just like we've been talking about, and I'm going to continue to talk about, is that form and function are closely tied together. What your form is, what your structure is incredibly closely tied to what your function is. So anyways, thanks for watching. Next time we'll be getting into the macromolecules.